Hello, I am Nur Nadia binti Abdurrahman with matrix number A168607. Today I will be presenting a project proposal entitled The Effectiveness of Baited Traps in Entomological Survey with special reference to the forensic scuttle flies. The supervisor for this project is Dr. Raja M. Zuha. Has anyone seen this kind of fly? Firstly, let me introduce the scuttleflies to you. The picture shown here is a scuttlefly species known as Megacelia spiracularis. For the first time, it has been recorded as forensically important species after it was found inhabiting copse indoor in Penang, Malaysia. It comes from a group of small flies, belongs to the family Phoridae, with over 400 species discovered worldwide. Scuttleflies are widely distributed and occur in different types of habitat, which makes them a suitable group for characterizing and comparing different ecosystems, including that environment in forensic cases. Another example is the cosmopolitan Megacelia scalaris, which can be found almost all parts of the world except the North Pole. Scuttleflies are known to be as one of the primary indicators to determine minimum post-mortem interval for indoors death due to their diminutive size, which is around 0.6 to 4 millimeters. Due to the small size, it can penetrate narrow space to reach corpse faster than other groups of forensic flies, such as Phoridae and Sarcophagidae. The occurrences of scuttleflies in human corpse have been discovered in forensic cases in Malaysia, but mostly restricted to indoors and confined spaces. Although scuttleflies have been featured in forensic cases as minimum PMI indicators, information about this fly is still limited. This is mainly due to lack of resources regarding collecting strategies that enable research can be conducted on species of scuttleflies. Previous studies have shown the efficiency of bait traps in attracting dipterans to determine the fauna of a region is based on the sense of smell and types of nutrition of flies. However, studies regarding the effectiveness of baited traps with special reference to forensic scuttleflies are scarce. Thus, by understanding the interactions between types of bait and traps on scuttlefly collection, it can provide practical solutions to focus on this species of forensic importance, especially when we want to study about the life history parameters that can be useful in forensic investigation. Therefore, we want to know what species of scuttleflies can be collected from different types of traps and baits. Is it possible to catch new species or new records? Do traps and baits influence the presence of forensic scuttleflies? In other words, does attraction to certain type of traps or baits are species specific? Lastly, does preference of scuttleflies towards different type of baits are based on sex? We believe this question can help us to further strategize collecting techniques of forensic scuttleflies that can be useful for forensic entomology application. From the research questions, we will conduct this research based on the main objective, which is to determine the effectiveness of baited traps in entomological survey with special reference to the forensic scuttleflies. To run this research, we will carry out separate experiments based on this specific objective. First, to assess species diversity of scuttleflies attracted to different types of traps and baits. Next, to determine the effectiveness of baits to collect forensic scuttleflies. And lastly, to determine the distributions of scuttleflies on baits according to sex. Therefore, we hypothesize that the effectiveness of traps and baits is attributed to species and sex of scuttleflies. Next, we proceed the presentation with the research design of this study. This research consists of two types of approaches, which are overt observational and experimental. We will be conducting the research at vicinity of UKM Bangi. The sampling instrument that will be used in the research involve two types of trap, which are Malaise trap and pen trap. The bait consists of decomposed animal tissue, modified bait, and control. Decomposed animal tissue will be prepared with 50 gram of cow liver, while modified bait will be prepared by mixing vinegar and brown sugar. The control group will only consist of traps without baits. The target population in this research is adult scuttleflies collected from all traps, and traps will be randomly placed at 6 selected grids at UKM Bangi. For measurement of variation, the traps will be prepared in 3 replicates. The whole procedure will be repeated minimum 3 to maximum 6 times. Figure 1 shows a schematic diagram on research design. As seen in the diagram, independent variable consists of 2 factors, which are factor 1 traps and factor 2 baits. The dependent variables in this research are total catch and diversity of the species from all traps. For the methodology of the first specific objective, which is to assess species diversity of scuttleflies attracted to different types of traps and baits. The experiment will be involving 2 factors, which are traps and baits. Traps will be placed at the sites and samples will be collected after 3 days. Adult scuttleflies obtained from Malaise traps and pen traps will be then preserved in 70% of ethanol. Next, identification of sample will be performed on the adult scuttleflies by dissecting the specimens and mounting them on slides. The species identity will be determined by using microscopy and taxonomic keys. The data of total catch per bait according to species will be collected and analyzed by using descriptive statistics. If there are any new species on records, they will be described according to taxonomic methods. The expected results for the first specific objective can be illustrated in Figure 2a by graphical display to show the number of scuttleflies collected based on its species for 3 types of baits, while Figure 2b shows taxonomic descriptions of new species or records. For the second specific objective, which is to determine the effectiveness of different types of traps and baits to collect scuttleflies, the methodology is as following. The same variables in the first specific objective will be involved in this experiment, which are the 2 types of traps and 3 types of baits. Adult scuttleflies will be collected 3 days after the placement of the traps. Next, the scuttleflies will be identified for the species. This procedure will give the total catch per bait according to species, and Shannon index of diversity will be calculated. To determine the significant differences of total catch and diversity of scuttleflies and the interaction between traps and baits on the dependent variables, we will perform factorial maneuver in R programming with 0.05 level of significance. The expected results for the second specific objective can be illustrated in graphical display showing total catch and diversity for each bait in Trap A and Trap B. Figure 3 simplifies how factorial maneuver analysis operates for multiple independent and dependent variables. As for the third specific objective, which is to determine the distributions of scuttleflies on baits according to sex. The experiment will be involving only one variable, which is baits. For sampling, the data from the first specific objective will be pulled according to type of baits. In data collection, the frequency of male and female scuttleflies collected from each bait will be recorded. For this specific objective, the analysis used is chi-square test of independence at 0.05 level of significance. Since we hypothesize that type of baits and sex of scuttle are unrelated, the expected results for the third specific objective can be illustrated in graphical display showing frequency of male and female scuttleflies from each bait. The Gantt chart shows the expected schedule for this research. This research is expected to start in June, and data collection is expected to be completed in August. However, due to COVID-19 pandemic, the research methodology might be modified accordingly to fit with the timeline. The following list shows the references that were used for this proposal presentation. That's all from us. Thank you and have a nice day.